Hello, everyone. In this presentation, we'll explore the regulatory requirements for peptide sameness studies, focusing on expectations from the U.S. Food and Drug Administration and Health Canada. Peptides are unique molecules that sit between small chemical drugs and biologics. Because of this complexity, regulators require strong analytical evidence to prove that a generic peptide product is truly the same as the reference drug. Let's look at why peptide sameness is so important and how it is evaluated. Peptide sameness is critical for both patient safety and therapeutic effectiveness. Even very small changes in the amino acid chain, such as oxidation or a missing residue, can alter the peptide's biological activity or potentially trigger immune responses. Because of this risk, regulatory agencies require strong analytical proof that a generic peptide has the same sequence, comparable impurities, and similar performance as the reference product. This makes analytical characterization the foundation of peptide generic development. Regulators treat synthetic peptides differently from typical small molecule drugs. Both the FDA and Health Canada often classify them as complex drugs, meaning they require more detailed characterization. In the United States, peptide generics are typically submitted through the ANDA pathway, while in Canada, they may be evaluated through new drug or abbreviated regulatory pathways. Regardless of the pathway, regulators expect clear evidence of API sameness, structural conformation, and comparable biological activity. The first and most important step is confirming the primary structure of the peptide. This means proving that the amino acid sequence of the generic peptide is exactly identical to the reference product. Scientists usually use advanced analytical tools such as high-resolution LC, MS, peptide mapping, and amino acid analysis. Peptide mapping is especially powerful because it breaks the molecule into smaller fragments and verifies each one, ensuring complete sequence accuracy. After confirming the sequence, the next step is evaluating the higher-order structure of the peptide. Even though peptides are smaller than proteins, their secondary structures, such as helices or turns, can still influence biological activity. Techniques like circular dichroism, NMR spectroscopy, and FTIR analysis help scientists compare the structural behavior of the generic peptide with the reference product. These studies help confirm that the molecule behaves structurally the same way. Another key regulatory requirement is impurity and variant analysis. During peptide synthesis, several types of impurities can form, including deletion sequences, truncated peptides, oxidized products, or structural variants. These impurities are typically detected using HPLC, LC, MS, and forced degradation studies. Regulators expect companies to identify and control these impurities and demonstrate that the impurity profile is comparable to the reference drug. Even if the structure appears identical, regulators still require proof that the peptide works the same biologically. This is demonstrated through bioactivity studies, which may include cell-based assays, receptor binding tests, or enzyme inhibition studies. These experiments confirm that the peptide triggers the same biological response and therapeutic effect as the reference product. In simple terms, it shows that chemical similarity translates into functional similarity. A key concept in regulatory evaluation is the orthogonal analytical strategy. This means using multiple independent analytical methods to confirm the same property of a molecule. For example, identity may be verified using mass spectrometry, peptide mapping, and chromatographic comparison. Using several complementary techniques increases confidence and helps regulators confirm that the generic peptide is truly identical to the reference product. In real pharmaceutical development, demonstrating peptide sameness can be challenging. One major challenge is the limited information available about the reference product's manufacturing process. Another challenge is detecting very low levels of impurities, which requires highly sensitive analytical methods. Finally, regulatory expectations continue to evolve as peptide drugs become more complex, so companies must design strong analytical strategies early in development. To summarize, demonstrating peptide sameness requires a comprehensive analytical approach. The generic peptide must show an identical primary sequence, comparable structure, controlled impurity profile, and equivalent biological activity. Regulators place strong emphasis on orthogonal analytical evidence, where multiple techniques confirm the same findings. Ultimately, these studies ensure that generic peptide medicines remain safe, effective, and therapeutically equivalent to the reference drug.